Biologically speaking, the digestive system is not an internal organ. It is a continuous, topological exterior tube running completely through the body. Its core objective is singular: to reduce structurally complex biomacromolecules into simple, uniform forms that the body can readily absorb. Achieving this requires two synchronized mechanisms: violent mechanical processing and highly specific biochemical cascades. This presents an immediate biological tension. The system must dismantle raw organic matter without accidentally digesting its own host tissues in the process. This diagram of the human digestive system maps the exact geographical roadmap from the oral cavity to the anus. As we follow this route, we will examine the specific structural modifications and chemical checkpoints along the alimentary canal. Digestion is a precisely engineered spatial processing pipeline. The location and timing of chemical releases are just as critical as the chemicals themselves. This gut cross-section reveals our four-layer processing architecture. The outermost serosa holds the tube together. Beneath it, the muscularis bands generate rhythmic peristalsis. The submucosa houses logistical supply nerves and vessels. Finally, the innermost folded mucosa is the active interface for chemical secretion and absorption. Every highly specialized organ we are about to examine is simply a targeted physical modification of this baseline four-layer architecture. The processing pipeline begins with initial mechanical destruction via human dentition. Our teeth are embedded securely in jawbone sockets, a structural arrangement known as the codont attachment, allowing them to apply massive crushing force to incoming food. The resulting masticated mass, now called a bolus, is driven forcefully down the esophagus via strong peristaltic waves. This anatomical diagram of the human stomach shows the bolus entering a highly specialized holding chamber, divided into four functional regions: the cardiac, fundic, body, and pyloric portions. Here, the baseline mucosal layer we saw earlier heavily modifies itself, diving deep into the tissue to form complex gastric glands. These glands produce divergent outputs. Parietal cells secrete hydrochloric acid, dropping the pH to a corrosive 1.8. Peptic cells secrete the dormant precursor pepsinogen. This safety mechanism prevents self-destruction. The zymogen remains inactive until it hits the acidic lumen, where it unfolds into active pepsin. Once activated, pepsin begins the aggressive preliminary cleavage of intact proteins, breaking them down into smaller proteoses and peptones. The stomach relies on strict environmental compartmentalization, separating the acid trigger from the enzyme's origin point to safely initiate protein breakdown. The resulting highly acidic semifluid, called chyme, exits the stomach through the pyloric sphincter and enters the C-shaped duodenum. This creates an immediate, severe physiological crisis. The acid must be rapidly neutralized, or it will severely corrode the delicate intestinal mucosa. This diagram maps the duct systems converging at the hepatopancreatic duct. Bile from the liver and gallbladder flows down the common bile duct. Simultaneously, the pancreas secretes alkaline bicarbonate juice to neutralize the acid, alongside a fresh payload of inactive enzymes. The duodenum effectively acts as the system's master chemical mixing valve, forcefully shifting the local environment from highly acidic to mildly alkaline to prepare for the final stages of disassembly. Here we encounter the autodigestion problem again. The pancreas has just released powerful proteases into the gut, but they are all dormant zymogens. The mucosal lining of the intestine secretes a highly specific enzyme, enterokinase, which initiates the first step of the catalytic sequence. Enterokinase converts inactive pancreatic trypsinogen directly into active trypsin. Active trypsin then triggers a runaway cascade, rapidly converting all the other dormant pancreatic zymogens into their active forms. These tables illustrate macromolecular breakdowns. Proteases cleave proteins, amylase reduces carbohydrates, and lipases target fats. The final step occurs at the mucosal boundary via succus entericus. Brush border enzymes convert intermediates into single monomers, amino acids, glucose, and fatty acids. This cascade ensures biological safety through a mandatory sequence of biochemical triggers, ensuring these potent enzymes are only produced once they reach the intestinal lumen. Absorbing water-soluble end products is straightforward. Simple sugars and amino acids easily diffuse directly into the bloodstream. However, lipids present a complex biochemical paradox. Fatty acids and glycerol are entirely insoluble in water, meaning they cannot directly enter the aqueous environment of the blood capillaries. The body's first workaround uses bile salts. Acting as biological detergents, these salts emulsify large fat droplets into tiny temporary structures called micelles. Inside the enterocyte cells lining the gut, the lipids are repackaged. They are wrapped in specific protein coats to form transportable spheres called chylomicrons. This cross-section details the highly specialized anatomical structure designed to receive them. Hidden deep inside the intestinal villi are central lymphatic vessels called lacteals. Lipid absorption requires bypassing the standard vascular highway entirely. The body is forced to utilize a dedicated lymphatic off-ramp just to transport its hydrophobic fuel. When the precise sequencing of this pipeline breaks down or encounters a deficit, the physiological consequences are immediate. Consider a mechanical failure. A physical blockage in the hepatic ducts causes toxic bile pigments to back up into the bloodstream, manifesting clinically as jaundice. This text outlines the systemic effects of a complete macronutrient failure, specifically analyzing protein energy malnutrition. It strictly differentiates between marasmus, a simultaneous deficiency of both protein and calories in infants, and kwashiorkor, which is a specific protein deficiency occurring despite a high-calorie diet. The entire process of sustaining human biology relies on strict structural integrity and sequential chemical precision. Rough mechanical crushing must adequately prepare the surface area to allow precisely timed zymogen cascades and highly specialized absorption vectors to function. The human digestive tract is an advanced, compartmentalized bioreactor engineered to systematically turn environmental chaos into usable biological order.